Okay. And the recording will be available after the event is over. So I also wanted to introduce Tucson Audubon for those who might be new to us. We are located in Tucson. Our mission is to inspire people to enjoy and protect birds as well as the ecosystems they inhabit. So we're excited to be giving this talk on something less avian, but certainly still related, of course. I am Kirsten Howe. I'm the Community Engagement Coordinator. I'm fairly new to Tucson Audubon, so hello to those of you who haven't met me before. Hopefully I'll get to see you in person sometime soon. Thank you for joining us. I also want to take a moment to thank those of you here who might be donors or volunteers, because your support is just so invaluable to us. So thank you for always coming out and supporting our efforts. And now to introduce our main event here, Jillian Cowles. Jillian is a microbiologist. She's also a photographer, and her photographs have been featured in many books, as well as in other publications. And we're just so honored to have her here to talk about something that I'm sure a lot of us are curious about, maybe a little scared of, and just excited to learn more about. So I'm going to go ahead and turn it over to Jillian. And like I said, feel free to throw your questions in the chat, and hopefully we'll have some time for those at the end.
Thanks, Kristen. Okay, so sharing, caring, and thievery, arachnid behavior and interactions. So let's start out with what is an arachnid? Well, first of all, it's an arthropod, which means it has an exoskeleton and multi-jointed segmented legs. Because it has an exoskeleton, that means it must molt in order to grow. So among the arthropods that we're familiar with are insects, like this ant, crustaceans, like this little sow bug, centipedes, millipedes, and chelicerates. Now, what the heck is a chelicerate? Well, chelicerates include arachnids and horseshoe crabs, and they are characterized by this structure here that you see on the scorpion. Now, in a scorpion, each chelicera consists of a fixed and movable finger. And these fixed and movable fingers work like little hands so that it can cut up its food into tiny pieces, masticate it, and introduce digestive enzymes into that food. And then it slurps down the pre-digested liquefied food. So you can see one chelicera here, and here you can see two of them. So they look almost like little legs. And in fact, from a developmental standpoint, they are appendages. But in terms of their functional standpoint, they are like little jaws. Now for a spider, each chelicera has a single naked fang. And these fangs in most species are hollow, and they have a hole at the tip so that they can introduce venom via the fang, just like a hypodermic needle. So among the arachnids are spiders. Here's the spider, okay. Scorpions, pseudoscorpions, Eurypygids, also known as Vinegaroons, schizomids, amblypygids, also known as tailless whip scorpions, palpigrades, also known as micro whip scorpions. These truly are micro. They're about 1 millimeter long, including the flagellum, the tail. Opiliones, also known as harvestmen or daddy longlegs. Solifuges, also known as sun spiders, camel spiders, or wind spiders. Retinuleids, that are known also as hooded tick spiders. And the ticks and mites. Now, ticks and mites used to be lumped together in a single group, but in recent years, they've been split out into two groups. One group is called parasitiformes, that includes ticks and some of the mites, such as this opilioacarid mite. And the other group is acariformes, which includes things like red spider mites, giant desert velvet mites, and most other mites that people think of as typical mites. So let's start out with the caring part with the Greenlink spider. So Greenlink spiders belong to the genus Pucetia. They are ambush hunters. They depend on their eyesight for capturing prey. This one has ambushed a little bumblebead fly that was visiting this flower. And they frequently do catch bees and other insects that visit flowers. You can frequently see Greenlink spiders lurking on prickly pear flowers in the spring as they capture their prey. We have two species of Greenlink spiders in the southwestern United States. That's Pucetia viridans, which has a highly variable color abdomen, and Pucetia longipalpus, which is a generally green colored spider. These are evolved primarily in the tropics and subtropics throughout the world. There are many species of Greenlink spiders. So Greenlink spiders have to protect their egg sac against potential predators, such as ants, beetles, and grasshoppers, as well as rodents and birds that might want to eat those eggs. So here's a Greenlink spider mother who has captured a katydid, and she's hanging out near her egg sac. And that egg sac actually has successfully hatched out some baby Greenlink spiders. They hang around on the outside of this egg sac for several days. They undergo a molt. You can see the exuvia from the little molts. And then they eventually balloon away to start their life. The presence of the mother is very necessary for the survival and successful development of these babies before they emerge from the egg sac. In one experiment where the mother Greenlink spiders were removed from the egg sac, the success rate of the egg sac dropped from 70% to about 10%. So the presence of the mother is very key to the success of the egg sac. So back in the 1940s, Captain Ernest Tinkham in the Sanitary Corps of the Army of the United States published an interesting little clinical note in the Bulletin of the U.S. Army Medical Department. And it was titled "A Poison Squirting Spider." And in this case, he was the doctor that saw a patient. The patient had been kneeling behind a motorcycle, checking something on the back of the motorcycle, and suddenly felt a sharp stabbing pain in one of his eyes. And he had the presence of mind to notice that there was a spider that appeared to have squirted something in his eye that had been on the back of his motorcycle, maybe behind the license plate or something like that. And so Ernest Tinkham drew some pictures of this spider and sent the pictures away for identification, and it turned out to be a Greenlink spider. Well, luckily the young man's eye did recover after a few days. But in 1984, Linda S. Fink published an account in the Journal of Arachnology titled "Venom Spitting by the Greenlink Spider, Pucetia viridans," in which she described that before spitting, a female shifts her weight posteriorly, lunging slightly forward immediately before or during the release of venom. So I thought to myself, well, this seems perfectly acceptable. I'll include this in my book. And I mentioned it in the book, and Rainer Felix, who is the author of Biology of Spiders, 3rd edition by Oxford University Press, read that in the book, and he contacted me and he said, there's just one problem. The hole in the fang faces the wrong direction. It faces inwards instead of forwards. So my husband and I undertook an investigation of how does the Greenlink spider manage to squirt in a forward direction. So what we did is we had some glass cut in 4 by 6 inch pieces. We put tape around the edges, and then we printed up this picture of owl eyes that we could hold behind the glass. And the first thing that we wanted to determine was, did the Greenlink spider actually squirt venom from a distance of a few centimeters? And sure enough, when we approached mother Greenlink spiders that were guarding egg sacs, we found that they did squirt venom on the glass. So here's a photo of some of the venom that was deposited on the glass. And you can see one droplet here and a little spray of droplets here. So I told Rainer Felix about that, and he said, well, I'm still puzzled. How the heck do they do that? So we started to get photos of them while they were actually in the act of squirting the venom. And here's a mother spider who is fiercely protecting her egg sac. You can see a stream of venom actually coming out of this fang. The other fang has already discharged and left all these droplets on this forceps. These are black plastic tips on a pair of forceps. And so a little bit closer up, you can see this liquid coming out and a little droplet hanging on the other fang. This particular Greenlink spider was in the actively moving when she scored it. So her aim kind of overshot the forceps tips. But you can see here the stream of venom, and her legs are actually blurred because she's moving. You can see that the forceps aren't squeezing her leg. They're not actually grasping it. So what we determined is that they start out, when they first see a threat approaching, they face it with their fairly good eyesight, because of course, as I mentioned, they're ambush hunters that use eyesight to pounce on prey that visit flowers. You'll notice at the bottom of their body is roughly in a straight line here. And then as the threat approaches, here are the forceps tips. What they do is they tip the cephalothorax, the front part of their body tips upwards. They extend the fangs outwards. And so the little hole on the wrong side actually is able to aim forwards by about a 45-degree angle from the angle of the body. So here you can see, here are the black forceps, and the spider is facing it. You're looking from the back of the spider. Here are the tips of the fangs right here. And in the next shot, bingo, she has raised up her body, extended her fangs, and she was able to shoot these streams of venom directly in front of her. So this was a really cool little study. It actually got published in the German journal Arachnid. Rainer Felix and I worked together on it. He did the SEM photographs of the fangs of the spider. And you can see that they can also shoot downwards here. So this defense of egg sacs is probably the most simple example of maternal care of young that you see in arachnids, and it's very, very common. So let's go to the next level, and that's in Vinegaroons. So Vinegaroons are named Vinegaroons because they can spray almost pure acetic acid out of the rear end from this little structure that works like a revolving gun turret. So they can shoot in any direction. They can rotate this little revolving gun turret called the pygidium and spray this acetic acid out. This is strictly used as a defensive capacity. They capture their prey with their heavy claws, their pedipalps, which you can see in this photo here. Now, Vinegaroons have a very complicated courtship. And when the male courts the female, he grasps the tarsomeres of her antenniform legs, the tips of her antenniform legs. These long, skinny structures are not antenna, they're actually legs, but they function like antenna. So I call them antenniform legs. So he grasps the tips of the female's antenniform legs in his chelicerae, and he nibbles on them. And they do a dance where they pull each other back and forth while he's holding on to her antenniform legs. And they can do this for anywhere from 3 to 5 hours. And at the end of that time, if they have decided that they're suitable mates, then he'll start to generate a spermatophore. And at that point, the female goes behind the male, embraces his abdomen, and during this 3 hours, he has to generate a spermatophore inside him before he deposits it. Then he deposits it on the ground and he leads the female over it. And so this is a spermatophore of a Vinegaroon, and she picks it up with her gonopore, and then she gets the contents inside her, partly because a male turns around and embraces her, and then helps to press in those contents with his pedipalps. So the entire courtship and mating of Vinegaroons takes approximately anywhere from 13 to 16 hours on average, which is a huge investment on the part of the Vinegaroon, but that's just the start for the female. That courtship takes place in the summer or fall, and in the fall, she digs a maternity burrow and she seals herself in that burrow. And during that time, she will not come out and feed during the entire 7 months that she'll be incarcerated in that burrow. Now, in the spring, she lays her eggs in a clear membranous pouch that she holds under her abdomen that I call a belly bag. And after about 5 weeks, the eggs hatch, and these little larvae climb onto their mother's abdomen. Sometimes she can produce her own weight in babies. There can be over 50 eggs in that pouch that can equal or even slightly surpass the weight of the mother. And what the babies do is they attach on the surface of the abdomen with these little sucker pads on their legs, and then they don't move at all for the next 5 weeks. And they undergo further development, tapping into these food stores in their fat little bodies. At the end of 5 weeks, they molt into nymphs, first instar nymphs. And so you can see them actually molting in here. Several of them have already molted and dropped to the ground. And here's one molting. Luckily, the little sucker pads of their old exoskeleton hold fast because they have to drop free of that. That means that the old exoskeleton is still stuck on the mother's abdomen. And eventually she gets kind of impatient with that and she'll scrape it off. And when she does that, the babies all crowd to the end of the burrow and make room for her. Now you'll notice that the babies here are darker already than when they first molted. So over the next month, they actually continue to darken, and eventually their dorsal surface will be black and their pedipalps will be red. During that time, they stay in the burrow, but at the end of that time, the mother sometimes starts leaving the burrow and she'll do some hunting on her own. And in this case, she captured a large cricket, brought it down into the burrow, dropped it on the ground inside the burrow, and then stepped back, and the babies all started tearing apart the cricket. Even after the babies emerge from the burrow, the mother continues to share food with them. So here's a little baby reaching right between the pedipalps of the mother as she's masticating and liquefying a cricket. The baby grabbed a chunk of this cricket mush and ran off with it. This was despite the fact that this particular mother had not fed for 7 months, had produced a large clutch of babies, and was absolutely famished when she captured that cricket. She still allowed her offspring to steal some of that food. And sometimes she gets actually mobbed by babies grabbing foods. Here's a chunk of cricket here, another one with a chunk, and several more helping themselves. So the most extraordinary case that I saw of Vinegaroons sharing food was this case. In this case, a mother had captured a large cricket. You can see the cricket. And then instead of going into her own burrow, she walked in the opposite direction and walked to the burrow of one of her babies. Now, the baby had already dispersed from the burrow and had dug its own little burrow. And it was in the opposite corner from the mother's burrow. So she walked over there and she reached her antenniform legs down into the baby's burrow, and up he popped and he grabbed hold of this drumstick of the cricket. And then they just stood there and tapped each other with their antenniform legs for a few minutes. Then she turned and walked to her own burrow with the baby still clutching the drumstick. She went down into her own burrow. And in that evening, they apparently ate the cricket, because in the morning there was a fat little baby on the ceiling of her burrow. The mother was fat and the cricket was gone. So what was really amazing to me about this was that this was in November, a full 4 months after the babies had dispersed out of the mother's burrow, and she was still actively sharing food with one of the offspring. So I was shocked by this, but honestly, I shouldn't have been, because any behavior that gives a selective advantage to the survival of your offspring will encourage those genes to be passed on for the next generation. In other words, if they can get past this critical stage of being tiny little babies, they have a better chance of maturing and passing on their mother's genes, right? So there's really nothing that weird about it. It's just that as a human, I didn't expect an arachnid to be caring for their offspring for 4 months. Let's go to the next kind of social behavior. This is Analozimos Arizona. This is a distant cousin of the black widow. And she's a tiny little spider. You can see that she's holding a fruit fly here. So she's not much bigger than the fruit fly. She's bringing this fruit fly to share with her babies, which are right over here. And in this photo, you can see the mother and that she's brought fruit flies to 2 groups of her little babies. And the babies are feasting on the fruit flies while the mother stands aside. So as the babies get a little older, they're able to hunt for prey themselves. But if they're still pretty small, what they do is that they find a large prey item like this fly that they can't overpower by themselves. They signal to their siblings, and their siblings come rushing over and help subdue the prey. And then everybody shares in the bounty. Now, this particular species of Analozimos is called a subsocial species because the babies disperse just before they reach maturity. Therefore, each colony only reaches about 30 spiders in size because they are the offspring from a single female. However, there are other species of Analozimos in the tropics which are permanently social. And what they do is generation after generation of spiders stay in the colony. And these colonies in the tropics can reach as many as 50,000 spiders in a single colony. You'll notice something interesting here. This is a little mirrored bug called Ranzovius Clavicornis, which is a kleptoparasitic bug that lives in the webs of these Analozimos. And there's as many as 6 of these little kleptoparasites in any one web, in any one colony. And even the adults stay in the colony and they steal food from the spiders. They feed on the prey. I'll be coming back to the idea of kleptoparasites later. Okay, so the next spider we'll talk about is Tinted Spiders. So Tintids are specialized for hunting flies, and some of them build their webs on the tips of vegetation. And just to give you some idea of how tiny this spider is, this fly is the same one in this photograph here. It's the same individual fly. So the entire web is smaller than the tip of my finger. And you'll see these at the tips of grasses. For if you were to take a closer look at this little web here at the base of the web, you would see that there's a male and a female Tinted that are cohabiting in the web. Now, some of the Tinted spiders like to build their webs on sunny boulders where flies like to bask. Tinted calcarata is one of those, and it also likes to build its webs on windowsills, which makes it kind of fun to watch. So here's a male Tinted calcarata, and it has these pointy little pedipalps, which makes it easy to recognize in the next photographs. So one day I was photographing some bombyliid flies sunning themselves on a boulder, and all of a sudden one of the small bombyliids started to struggle. And I took some photos of it, and I didn't even realize what was going on until I got it up on my computer screen later. Because I could see that a spider was capturing the fly, but what I didn't realize is that there were actually 2 spiders. There was a female, and then here's the male behind it. You can see that pointy little pedipalp here. And after they captured the fly, then they shared the bounty. Here they are feeding on the fly. So in this case, I think that the Tintids cooperatively kill prey. Adults do, because any one of these spiders might not have been able to overcome a fly by themselves. They are quite tiny. They're not the only ones that share prey, though. This cellar spider, this male cellar spider, had captured this Pseudo Scorpion. And the female cellar spider came over and she was indicating that she wanted to feed on that scorpion, and he allowed her to feed on it. They both fed on the scorpion for 24 hours. So that might seem very chivalrous and altruistic, but when you think about it, he probably had a much better chance of mating with a well-fed female And plus, a well-fed female produces a good clutch of eggs that passes his genes on to more individuals. So it's a case where natural selection would indeed reward a generous, altruistic male when he shares food with the female. Now, so far we've been talking about sharing and cooperation within a species. Now we're going to have some examples of arachnids interacting with other species. So in this case, this is a furulithid spider, and it lives in the nests of chromatogaster ants. Nobody really knows what these spiders do in this nest, whether they are parasites in the nest feeding on ant larvae, or whether they feed on some of the other inhabitants of the ant nest. For example, there could be collembola or beetles, or a little myrmecophilic cricket living in the nest. And here you get a better view of this furulithid spider. And here's a little bristle millipede living in that chromatogaster nest as well. So it's possible that this is a parasitic relationship of the spider in the ant nest, or it could be a commensal relationship, or it could even benefit the ants if the spider is killing some kind of an animal that might prey on the ant larva. Nobody really knows, but these spiders typically are found only in ant nests. Another arachnid that has a relationship with other arthropods are pseudoscorpions. Now, this is a tiny little animal. Its body length is only about 3 millimeters. And what pseudoscorpions are really famous for is hitch-hiking or foracy on other arthropods. So this is a stratiameid fly, and the pseudoscorpion was hitching a ride by grabbing the leg of this fly. And the fly was probably on a rotten cactus, like a rotting saguaro, and then it flew over to get a little drink from the edge of a stream, and I got this photograph. But if it had flown from there to another rotting saguaro, that would have given this pseudoscorpion very good habitat for hunting little arthropods. And some pseudoscorpions hitch rides on cerambycid beetles. Now, many of them will hitch a ride under the wing covers, these hard elytra, and you can't see them. But this particular species rides on this cerambycid beetle where you can actually see them. And they seem to travel in little groups like this. There's no way that these little pseudoscorpions could travel from one tree to another tree easily on their own. They're very, very tiny. And their little legs, it would take them forever to get somewhere. But by riding on the beetles, they can disperse to new habitat, to a new tree, and then live under the bark of that tree and hunt for little arthropods. Probably the most famous case of foracy or hitchhiking in arachnids are mites on various beetles. And these are mites that ride on a carrion beetle. Now, this carrion beetle, I offered it some suitable carrion in the form of moist canned cat food, and it started to feed on that cat food. And as soon as it started feeding, all the little mites dove headfirst into the cat food and started to feed as well. But as soon as the beetle was finished, they all climbed on board again, and not a single mite was left behind. So they're very closely attuned to the signals of the beetle in terms of what the beetle is going to do. Now, what these carrion beetles do is if a mated pair of carrion beetles finds a little corpse out on the ground somewhere, a little bird or a little rodent, what they'll do is they'll bury that little corpse and they'll lay an egg on it, and their larvae will feed on that carrion. At the same time that they do that, these little mites will dismount from the beetle and they'll lay eggs on that carrion as well. And you might think, well, why on earth would the beetle allow these little mites to ride around with it if they're going to compete with its offspring for that resource? But in fact, what people think that these little mites might be doing is that they might be predating or hunting the larvae of flies, little maggots and other kinds of beetles that might compete with the carrion beetle's larva for that little carcass. So they might be repaying the beetle for the ride by eliminating competitors for that resource. Okay, let's talk about black widows and kleptoparasites. So everybody knows what a black widow looks like, and this is Latrodectus hesperus, our western black widow. And typically what black widows do when they're incubating an egg sac is that they'll build a tubular silk retreat. You'll have to take my word that this is a tubular retreat. And they'll keep their current egg sac in that tubular retreat where they can protect it easily. And the ones that have already hatched out the eggs, she just pushes out and discards just outside of the retreat. So normally this is what you see with a black widow, is the egg sac up in her silk retreat. But you'll notice something really weird here, is that there are two black widow egg sacs way out in the periphery of this black widow web. And you'll also notice something else really strange. There are 4 little spiders in this black widow web. There's a female here, a gravid female here, another female here, and a male here, all adult little spiders. And these little spiders are Argyrodes pluto. And Argyrodes refers to the silvery area on them, which the word for argentum in Latin means silver. So almost all Argyrodes have a little bit of silver on them. So on occasion, on exactly 2 occasions, in all the time that I've studied these things, I've seen Argyrodes pluto gleaning small abandoned prey in the web of the black widow, such as this little moth, and they'll feed on that. But much more frequently, what you see is something like this. Here are 3 adult Argyrodes pluto in a black widow web. Here's the black widow egg sac. Here's an adult female pluto, another one, and a male. And the male has a little white something that he's feeding on. And if you look more closely on that little white something, that white something is a baby black widow. Believe it or not, baby black widows are almost pure white when they first emerge from the egg sac. When he was done with that, he went back for seconds. And you'll notice that he has just a small hole in that egg sac. Here are the eggshells from the baby black widows that have hatched and are undergoing further development in there. And he pulled out those eggshells and he's trying to get another baby to feed on. And when he was done, the females took turns pulling out baby black widows and feeding on them. And here you can see a better view of what a baby black widow looks like when a female pluto is feeding on it. So most Argyrodes throughout the world live in the webs of orb weavers. And what they do is they steal wrapped prey bundles from the orb webs. So my question is, why on earth does Argyrodes pluto live in black widow webs here in the desert Southwest instead of in orb webs, the way most Argyrodes around the world do? So I looked at 3 different species of orb weavers. This is Argyrope trifasciata. And one of the problems with this kind of spider, although it keeps a fairly permanent web, it doesn't take it down on a daily basis the way some orb weavers do. So in theory, that might be a pretty good place to live. But the problem is that these only become large towards the latter half of the monsoon season. Until the latter half of the monsoon season, which is like August and September, these spiders are pretty small and they're wrapping only very small prey items. And then when they produce an egg sac, they produce just a single one, which they do in a secluded area, and then they guard it until they die. Neoscona oaxacensis, the western spotted orb weaver, would be another candidate, but these take down their webs on almost a daily basis. And once again, they're really only a big spider towards the end of summer, which wouldn't allow the Argyrodes pluto very much time to complete their life cycle. And then we have Metaphyra, which is a fairly common, abundant little orb weaver, but they're small. They don't have very large prey bundles to steal. And when they produce egg sacs, they tie them together with a lot of silk. So it would be very hard to steal these. And their prey bundles wouldn't be very significant. Whereas the black widow what the black widow does is produce multiple egg sacs. She'll produce one after another in the course of a summer if she gets enough food. A really well-fed black widow can produce as many as 9 egg sacs in a single year. And in addition to that, the black widow's web is more or less permanent. It stays in one spot. And in addition to that, the black widow can live for 2 or even 3 years in a really good location, which provides a long-term benefit for the Argyrodes pluto. So the Argyrodes pluto has switched from stealing prey bundles in orb weavers' webs to stealing egg sacs from comb-footed spider webs, specifically the Western black widow. Now you'll notice that this Argyrodes pluto is feeding on a baby black widow as another one has successfully exited the egg sac. And the neat thing about these Argyrodes pluto is I think you're seeing some co-evolution taking place here, because instead of simply ripping a big hole in the egg sac, which would compromise the development of the remaining eggs and embryos, instead they chew just tiny little holes in them. And the remaining babies continue to develop successfully and emerge from the egg sac. And in some cases, you can see as many as 70 babies successfully emerge from an egg sac that the Argyrodes have been feeding from over several days. What's interesting though is that these babies hit the web running. Instead of hanging around on the egg sac the way most baby spiders do, they come out of the egg sac and they disperse immediately away from the maternal web. They're gone within like 2 hours. And I did see an Argyrodes pluto briefly pursue some of these babies, and they were the baby black widows easily outran the Argyrodes pluto. But I think the presence of the pluto in the web has selectively gone over evolutionary time. The babies that dispersed rapidly from the web had a selective advantage because of the presence of these tiny little Argyrodes pluto that were predators in the web. So I call this precocious dispersal. Now, how did the Argyrodes pluto manage to steal that egg sac from the very protective mother spider? Well, they wait for her to wrap a large prey item like the Carrion Beetle when she's several inches away from the entryway of her silk tunnel retreat. At that point, the little plutos spring into action. They rush to the silk retreat, and a few minutes later, they appear with the black widow egg sac. They manipulate it with their legs and by attaching silk and carrying it off to one side of the entrance first. And then they attach silk and move it in increments away from where the black widow normally goes. They seem to know exactly where they can move that egg sac, where the black widow rarely goes. And in one case, though, I saw something really extraordinary. Normally, they will transport the egg sac this way by attaching silk. But in one case, the little pluto female simply pushed the egg sac out of the entrance of the silk retreat and allowed it to fall on the floor of the porch. And then she zipped down, found the egg sac, and suspended it a little bit outside of the black widow web. And there were several things that were really extraordinary about this. The first thing that seemed extraordinary is it had all the appearance of planning. She pushed the egg sac out. It totally fell on the floor without being attached to silk. And she knew exactly where to find it. And then the fact that she suspended it outside of the black widow web, it almost looked like she was cheating in a way, because these Argyrodes pluto normally live in little groups of 2 to 4 adult Pluto spiders sharing a living space in the black widow web. And normally, if any one of them steals an egg sac, it shares it with everyone else. So possibly she was cheating by suspending it outside of the black widow web. But interestingly enough, 2 nights later, I found her dead on the floor of the porch. So maybe when she leaves the black widow web, she's leaving a relatively protected area. So normally they suspend them with silk and move them in increments, a distance away from the entryway of the silk tunnel retreat, but they're still within the black widow web normally. You'll see these light colored objects in the background here. This little female was moving that stolen egg sac to an area where she liked to hang out. And these are her own egg sacs that she had produced in the past. So after feeding on the stolen egg sac for several days, the Argyrodes Pluto are enormously fat and ready to produce their own egg sac. So they start out with building a little goblet shape here, a little cup shape. And then they tip that on its side and start extruding their eggs into that. And they do it all in one big push. So it only takes them about 1 to 2 minutes to lay all their eggs. They do it all at once. You can see this deep dimple in the back of her abdomen as she's actually laying her eggs here. And at the end of this process, she has produced an enormous volume of eggs when you compare it to the size of the spider. So this is a big investment on the part of the little Argyrodes. Then she wraps the entire outside with this sort of flocculent silk. And then she puts layer upon layer upon layer upon layer of very fine silk, including building this little neck where the baby spiders will emerge. It takes her about 4 hours of constant work to build the egg sac. And at the end of that time, she's exhausted and she rests with her long front legs lying across the egg sac as she embraces this egg sac. Now she has to protect the egg sac because there's a little egg parasitoid wasp that is intent on gaining access to that egg sac. Arachnopteromalus dazzes will parasitize the eggs of this little spider. So she uses leg sweeps to try to shoo away the wasp. And then if the wasp lands on the egg sac, she'll actually actively pursue the wasp, chasing it around on the egg sac. She grabs it by a leg, and then she flings it. So this active defense of her egg sac against a parasitoid wasp, I thought was really extraordinary. I had never seen any documentation of that before. And the little wasp was very persistent though. And I watched this going on for about 40 minutes until I finally couldn't stand it anymore. This emaciated little frantic spider trying to defend her egg sac. So I finally collected the wasp so that I could get it identified and to give the poor little spider a break. Now, before this Argyrodes Pluto lays her eggs, she looks like a little sparkly Christmas tree ornament, enormously fat. But at the end of laying her eggs, she has a slightly shrunken appearance. And so at that point, she's ready to go steal another egg sac. So here's another theft of an egg sac. She's moving it away from the silk entryway. Here you can see this tubular silk entryway to where the black widow had been incubating her egg. And this is on an old tire, an old bicycle that I had parked on my front porch under a porch light. So the black widow web was in a really ideal spot for the black widow to capture lots of prey. But the size of the tire here, you can see that that gives you some scale of this little Argyrodes moving the egg sac. And you'll notice a little black something on the bottom of the egg sac. Now, during the time that the black widow wraps prey, almost every time that she was wrapping large prey, she would stop in the middle of it and rushing back to her silk retreat. And in this case, she spent about 10 minutes in there, but she was too late because the little Pluto had already stolen the egg sac and apparently had moved it to a spot that the black widow wasn't aware of. And by the next morning, here's the little Pluto with her egg sac. You can see how far away she had moved it, but it's still within the black widow web. And here's the top of the silk retreat of the black widow. And at that point, she started to feed on the contents of the egg sac. Now, one of the questions that came up was, why on earth does a black widow tolerate the presence of these little kleptoparasites in her web? And that's a really good question because I had never seen that question posed in any of the papers about kleptoparasites in the webs of spiders. Why do they tolerate them? Well, you know the old story of, oh, well, first of all, I'm going to talk about one other thing, is that the little black something that was on the outside of the black widow egg case, if you get a closer look on this, it's a little wasp. And this is Philolema latrodecti. And what these little wasps are egg parasitoids that specialize primarily in black widow eggs. And it's not alone in competing for that resource. This little Pseudogarix signatus fly, what it does is it lays its eggs on the outside of the black widow egg sac, and its maggots burrow through and devour the eggs. So the little Argyrodes pluto has a lot of competition for the egg sacs. Okay, so back to the question of why does the black widow tolerate the presence of those little kleptoparasites? Well, there's an old story about how to safely hike in grizzly bear country. And the joke goes that the way that you hike safely in grizzly bear country is that you go with a hiking partner that runs a lot more slowly than you run. And the idea is that if a grizzly is chasing you, you don't have to outrun the grizzly. All you have to do is outrun your hiking partner, right? And the grizzly will catch the hiking partner. Well, I think that this is the grizzly in the case of the black widow. This is a pirate spider, Mimetis. And what these spiders do is they hunt and kill orb weavers and comb-footed spiders, such as black widows and Argyrodes pluto. And I believe that possibly the black widow tolerates these Argyrodes pluto in its web because the spiders that tolerate them have provided low-hanging fruit for these predators. And possibly the predators will pick off the little Argyrodes pluto instead of killing the host spider, the black widow. It's certainly a possibility. Okay, let's look at one more pair of kleptoparasites and Tinted spiders. So Tinted Spiders is another cousin of the black widow. It's a comb-footed spider, and it lives along riparian areas, the walls of riparian areas. And you can see these cottonwood leaves. And what it does, instead of making a silk tunnel retreat, is it fastens together some leaves, or it ties a leaf shut like this, and it lives in this little shelter that builds with the leaf. And these are just hanging there in the middle of a web that looks very much like a black widow web. So what I found in the Tinted webs are these little spiders. At rest, they look like debris. Here's the egg sac of this little Neospintherus bavokiveri. When they're not at rest, you can see that they look more like a spider. The female looks like this. They can be highly variable in color. And here's a male. And the reason why I'm showing this male is because you'll notice on the far side of his body, he has these two long legs, but on the near side, he just has these two stumps. So he somehow lost these two legs. And keep that, file away that little incident for future reference. Now, normally, Neospintherus bavokiveri had been only documented as a solitary araneophage or spider hunter that had been found in the webs of Philopinella spiders. Now, Philopinellas are very small spiders that completely lack venom. They're in the only family of spiders that has no venom. And what these little Philopinella do is that they're facultatively communal spiders. So they each have their own little web, but it's immediately adjacent to or above or below another little Philopinella web. So you can see this whole little community of them, almost like a little apartment building of Philopinella spiders. And what the Neospintherus had been documented as doing is sneaking into the webs of these spiders, and as a solitary spider hunter, it would simply kill and eat these. However, in the webs of Tinted I found that they were kleptoparasites. They were gleaning little prey items from the host spider, things like ants and leafhoppers. And there were as many as 2 or 3 dozen of these little spiders living in each Tinted web. Also in the Tinted webs, I saw our old friend Philolema, which was an egg parasitoid wasp. Now, one other thing that's really interesting here is that you'll notice that here's an adult Tidaran Sisyphoides with her egg sac guarding it in this leaf shelter. And you'll see this odd little spindle-shaped cocoon in her web. And when I collected some of these little cocoons, what emerged from them were Zatypoda. Now, Zatypoda wasps are ichneumoniid wasps. And what ichneumoniid Zatypodas do is that they lay their egg on the dorsal surface of a Theridiid spider. And the larva, this is a wasp larva, feeds on the blood of that spider over a period of time until the wasp is ready to pupate. At that point, it kills the spider and it spins a little cocoon right in the web of the spider. The fact that I was finding the cocoons right next to obviously alive Tidaran makes me strongly suspect that the Zatypodas that were emerging from the cocoons had used the Kleptoparasitic Bavokivri, the Neospintherus, as the host. So possibly this was analogous to the black widow tolerating the Kleptoparasitic Argyrodes pluto if those Argyrodes pluto served as prey for the mimetis. Maybe it's low-hanging fruit for parasitoids and predators. So also in this same environment, you'll see the cottonwood leaves were Latrodectus hesperus webs. And you'll notice this curved cottonwood leaf here, this bent stem. I'll refer to that exact same leaf in the next slide. So here's that same leaf in the background. And in that same web, there are 5 Neospintherus Bavokivri. So you'll notice 1, 2, 3, 4, 5. There were actually 2 more in addition to that in this same web. And also in that web was a lone little Argyrodes pluto guarding her egg sac. But you'll notice something really odd about this one. She's missing those 2 really long front legs. So somehow something had happened to her front legs where some kind of a aggressive interaction, she had to drop a leg, actually 2. And I suspect it's the presence of the Neospintherus Bavokivri in the web that caused this leg loss. I never saw any Argyrodes pluto that were missing legs if Neospintherus were absent. And the fact that the Neospintherus are documented as being spider hunters makes me think that they can be pretty aggressive with other spiders. So this little Argyrodes pluto was having to deal with all those little Neospintherus on her web. So in a single black widow web, you can have the black widow, maybe 1 or more Argyrodes pluto, Neospintherus Bavokivri, Philolema latrodecti, Pseudogarus signatus, Arachnoptera malus dazes, Zatypoda, and let's not forget the pirate spider, Mimetis. So what this brings to mind is the idea of Indra's net. Now, Indra's net is a metaphor that popped up in the literature in ancient India. And what Indra's net was a spider web where the silk connected all things in the universe. And the web was hung with dewdrops, and every dewdrop reflected the entire universe. And I feel that the metaphor of Indra's net is reflected in modern day terms like the World Wide Web and the internet, which uses that same metaphor of a spider web to interconnect everything. And what I like to finish with are a couple of ideas. First of all, we're all interconnected and the world is very complicated. The second thought is that in many ways, natural selection is portrayed as being a knockdown, drag-out fight of competition, where nature is red of tooth and claw. But I feel that frequently, if you look, you'll see cases of tolerance, cooperation, and even altruism commonly in nature as well as the competition. Nature really doesn't care. Natural selection doesn't care. Whatever works, works. And so I think it's important to remember that sometimes cooperation and altruism may have a selective advantage, even more than competition and drag out fighting. And I think that that's a really nice thought to leave with. So that's the end. And I'd like to remind people that this book is available in the Tucson Audubon gift shop. A lot of the material that I presented in this talk is not in that book because it's my most recent research, but there's tons of other material in the book. So if you're curious about more stuff to do with arachnids, you can look in that book. Okay, any questions?
Jillian, thank you so much. That was fabulous and interesting. We do have a couple of questions. Just a note, I dropped the link to the product page on our shop in the chat there for anyone who's interested. So just a couple questions that came in while you were speaking. So the first one is from Joanne. She says, what other spiders make a messy 3D web like the Black Widow? She says she has lots of messy webs, but rarely sees a spider.
Okay, there's quite a few actually that do sort of three-dimensional irregular webs. One would be Dickwitia, the shrub spider that I was talking about with one of the people before the talk started. You'll see those in like bushes, primarily bushes and prickly pear cacti, things like that. Another one would be our cellar spiders. They create rather irregular three-dimensional webs. And there's quite a few others, but basically all the Therideids, all the comb-footed spiders built little irregular messy webs. And there's a variety of different web shapes, like sheet webs that things like Agelenopsis, the funnel web weavers build and stuff like that. So, you know, there's many, many variations of web shape and design. And it's very helpful when you're trying to identify a spider to sort of take a look at that web, because when you look them up, that will be one of the characteristics that will help you identify them.
Makes sense. The next question comes from Joan. This is also something I'm wondering about. She says, the photography is amazing. What camera setup do you use?
Okay, I use a Canon camera. I've gone through 3 different bodies of Canon in the past 15 or 20 years. I started out with an EOS Rebel. Basically, you want a camera that's a single lens reflex camera that will allow you to interchange lenses, because what I use as a special lens for really small things like the Argyrodes Pluto is an MPE 65 millimeter macro lens. And that's a really, really nice macro lens, but it's only good for really small stuff. And then I also use a 100 millimeter macro lens and a compact macro lens. So I use a variety of lenses. I use a Sigma ring flash that fits, the light part of it has 2 lights. It's not actually a ring anymore in the light. It's 2 lights that fasten to the front of the lens. And I can program each of those lights manually and independently of each other. So I strongly recommend having a flash, because without that, you won't be able to stop action. And so that's the basic equipment, is a camera that gives you the chance to manually select your settings, and flash. And that should pretty much, and some good macro lenses.
Wow, thanks. That's really, I mean, the results speak for themselves, really incredible. So we also have a couple of questions about the Vinegaroons in particular. One from Marley about how long they live in the wild, and another one, also from Marley, about just more about the environment that you observed them in. Were you working with them in a lab, and what size I guess, lab do you use to allow the burrows of all the individuals?
Okay, In the Vinegaroons, actually, I did work with those primarily in terrarium setups. However, if you're interested in Vinegaroon natural history, Justin Schmidt, author of Sting of the Wild, did a lot of research on Vinegaroons, and he has a couple of publications. There's one publication that should be coming out very close to this time in the Journal of Arachnology, and another one that will be coming out or already came out on Vinegaroon natural history. He has studied the life expectancy of Vinegaroons, and to answer that question, in the laboratory, he found that if they were fed well, they would molt once a year, but no more frequently than once a year. In other words, you could feed them as much as they can eat, and they still only molt once a year. If you don't feed them abundantly, it might take more than 1 year, like 2 years between molts. They go through about 5 molts before they reach adulthood. So it takes them about 5 years under good conditions to reach adulthood. And it could be longer than that if they have lean years. And then once they reach adulthood, they can live about 3 years beyond that. So their maximum life expectancy would be somewhere around 8 or maybe 9 years. But in the wild, who knows exactly? But in the laboratory, under conditions that could be controlled, that's what he found. My setup used a cutaway burrow where I used a glass terrarium and then built a clay burrow for the female Vinegaroon that had a cutaway area that sat close up against the front of the tank, on the inside of the tank. And then I covered the front of the tank with black construction paper, and I could peel that back at night and use a red light to observe behavior. So at night, the Vinegaroon could behave under fairly normal conditions, and I could see what was going on in the burrow without it disturbing them. You have to use something like clay that can be fired, because if you don't fire the clay, she will mess around with that clay and smudge the inside of the glass. And it also has to be something like clay or plaster of Paris, because moisture has to be able to permeate through there and give humidity for the babies inside. They actually drink some of the water that forms this condensation inside the burrow. So if you use something that's non-permeable, they'll dry up. So you need something like clay, fired clay that has not been glazed, or plaster of Paris for building the burrow, and with a cutaway front up against the glass. And that's basically it, observing them at night. And I'll say this, the Argyrodes Pluto, I observed that on my front porch, but that was basically a natural setup. They moved there on their own. The Black Widow moved there on her own. They found the front porch to be a good habitat, but it didn't mean spending a lot of all nights out there observing, because it only takes about 3 minutes for them to steal an egg sac. And so you can be waiting all night, and you have to be ready to photograph that when they do it. You have to be awake and alert and photograph this tiny little, roughly 5 millimeter long spider as she's moving around, stealing the egg sac or laying her eggs or doing other things. That almost all of that happens at night. So.
Wow, and it's just incredible you were able to capture all of those great photos, considering, how easy it would be to miss all the action.
Long nights, and I'm getting way too old for that. I can
only imagine. So we appreciate your sacrifice so we can see the end result. So just another another question about whether or not you would be comfortable sharing your email so people could ask for spider IDs.
Sure. And it's Jillian at mindspring.com. So JILLIAN at mindspring, MINDSPRING.com.
Yeah, I can put that in the chat.
And I'll try to get back to people with their questions. Sometimes I get really busy and it might take a while. And if I haven't gotten back to you within a week, then you can bug me again, because I just get really busy once in a while.
Yeah, that's great. Well, thank you so much, Jillian. This was so fascinating. I think I can speak for everyone to say that I just learned a ton. And we really appreciate you spending this time with us, teaching us about arachnids. And we hope that, we have a future collaboration, maybe on your next book. That would be wonderful. And thank you so much to our audience for sticking with us. I'm going to throw in the chat just the link to our upcoming events in case there's any other events that you'd want to sign up for. We really appreciate you being here. And thanks again to Jillian. This has been really wonderful.
And thank you, Tucson Audubon, for having me. And thank you for the folks that listened in, because arachnids tend to be among people's least favorite creatures. And I feel this intense gratitude for being able to share my view of them, because I get to see things that have given me a really different view of these little creatures. And I feel like they deserve a second look. So
absolutely, I totally agree. So thank you for providing that for us today.
Thank you very much. I loved it. And now I guess I got to put your book on my Christmas list. And I love all my little arachnids. I wish they would come eat more of my damn crickets.
Yeah, they do what they can. My cellar spiders, I always allow quite a population of spiders to live in my house. And every year they pick off a couple of kissing bugs. So they really earn their keep. Because I'm not too crazy about kissing bugs in my house. So if you want to have a little help wiping out the stray kissing bug that might get in your house, allow the cellar spiders to live in there.
Yeah, great free pest control, right?
Yeah.
All right. Thank you, everyone.
Well, thank you.
Bye.